So hi everyone, hi, hello and welcome. Microbehunter here, I'm Oliver. Okay, it seems to work. Okay, just let me check. Yes, I think I'm now online, and I'm not only online once, but two times, as a matter of fact, because for the very first time I'm trying to stream both horizontally and vertically. Yeah, and I'm just trying to sort out all of the technical stuff first. Yep. Okay. Yeah, I see that several of you have already joined in. So I'm happy that we can do a little bit of microscopy like every week together. Yeah, as I mentioned, I'm experimenting around a little bit. You're now basically seeing both, or you can see in two different streams, either the horizontal version, but I'm also trying to do some vertical live streaming. So there is also a link where you can try that out. And the layout is a little bit different, and I just would like to see how this works out. Okay, so yeah, let me quickly check how this works. Yep, it seems to be okay. I see that I can see myself both in the horizontal and at the vertical one, and I hope that the computer is not going to be overloaded, that the CPUs are not going to be overloaded. Okay, yeah. First of all, as always, hello around the world, to Stereo Austria, hello to the Netherlands, United States, Vietnam, Scotland, Netherlands, UK, yeah, Florida, New Jersey, yeah, and North Carolina, Rhode Island, UK again, LA. Wow. Okay. Yes, and we're gonna see how this actually works out this time. And because it's a little bit experimental, I decided to do something today a little bit, traditional again, because maybe there are going to be a few new viewers. I've not collected, but basically I've put out a whole bunch of microscope slides, which I consider to be one of my more favorite ones. Yes, not all of them are made by myself. Some of them are actually quite are commercial slides, but they look kind of nice. And yeah, I every now and then return back to those, and I simply also would like to share them with you. Of course, this gives us also a little bit of an opportunity to chat about other things that you might want to talk about. From an organizational perspective, it's like this, that I'm going to now, I can see the chat from the horizontal, from the traditional horizontal live. There is also a chat in the vertical one, but I am not going to be able to join those two chats. So if you are viewing the whole thing in the vertical one, well, actually, I can switch back and forth a little bit. So maybe this does work. I'll have to see. Okay. Yeah. The monkey tail. Yes, I got it here as well. So I'm going to just do it in a very how do you say, traditional way. If you want to have a look at anything else that I've got here, of course, feel free to request this as far as I've got it. Otherwise, we're just going to look through the whole thing, and because every week there is. Even if I'm looking at static images, I do want to show you something that I just discovered 10 minutes ago, and that is the following. Just not a, how do you say, not a permanently mounted slide, but a temporary slide from a flower pot, flower pot water, and there was plenty of life in there as well. Okay. So that's the thing. There's an interesting comment here. Just a second. I'm also switching back and forth a little bit on whether I'm actually also able to see whether the vertical one works. So I'm still, yes, it still works. Okay, so that's also always something that I have to now check. Yeah, I just wanted to show this to you as one of the things that which I just discovered a little bit at this flower pot water, which looked pretty unappetizing, and I found also a couple of ciliates, mostly nematode worms. I have some rotifers. Yeah. Here, plenty of worms as always. Just wanted to share that with you. Some interesting, more or less interesting ciliates, of course. Yeah. So that's something that I think we all like to do. We occasionally like to explore things. However, those slides, even though there is some nice, interesting movement going on here, of course, they don't quite look always as pretty as some other ones, especially plant cross sections and so on. And for this reason, I decided, okay, let's have a look at a few of my my other favorite slides that I have, and then let's take it from there. Okay. So yeah. Let's have a look. I just want to show you first something that I tried to make several years ago. It's already over 10 years ago. This is a so-called paper-covered slides. This one is also a nice one. Many of you already know that. That's the veins of a leaf. Every fall, I like to put some, I'm preparing some leaf veins, and yeah, they look quite nice. And I made a permanently mounted slide of that, and I'm going to, of course, also turn off the arrow. And usually they look quite nice, under low magnification. And look what's going on here. This is now dark field, and this was quite a long preparation. If maybe some of you do remember this in a few months ago. Yeah, I had to boil the leaf, and then I had to strip off all of the loose cells. Yeah, and for this reason, yeah, this is also one of the my favorite ones, because it kind of shows a little bit the beautiful patterns of nature. Yeah. And it's also a paper covered slide. I did some experimentation with paper covering slides, because antique microscope slides sometimes are also covered with paper. Yeah. Yeah, and there's an interesting comment already that basically in the old, apparently, Jesse, ever smaller, red, green, and yellow paper antique slides should be handled with gloves and surfaces cleaned after contact. They often have arsenic in the inks. Now, that's quite interesting. Didn't know that. Okay. As a matter of fact, it is something generally that one has to be aware of when dealing with antique items. I mean, in the past, they also used, for example, radioactive uranium glaze for ceramics. Yeah, people didn't know that radioactivity was a problem at that time. So it is indeed the case that 100 years ago, the whole issue concerning safety, people simply didn't know many things, right? So it doesn't surprise me that maybe they did use. I'll show you a couple of my antique slides that I have as well. But I'm going to just go through those here just to share with you a little bit. So this is one that I've made basically myself, right? So, and it's one of the things that I like, because this was a little bit an experimentation on making them paper covered. Yeah, this one here, with my school students back in 2006. So this was already 20 years ago. I visited South Africa for an exchange program. This is a little souvenir from South Africa. It's the feather of an ostrich. And because I also would like to always check every now and then to see how well they have actually stood the test of time. Because I always want to know whether they're still sufficiently stable. This is a little bit too. We see already there's some bubbles here. Yeah, but this here is the feather of an ostrich, and because I've got DIC, so I'm just gonna look at it in bright field. But it looks a little bit like this feather has survived. Yeah, the past, well, in this case 16 years, because I made the slide a few years after I visited South Africa. So that is the feather of an ostrich, and not all specimens look very nice. Because sometimes there's some preparation problems, but still they do have some kind of a, how do you say, sentimental value, if you want to see it like that. Yeah. Let me check again. Yep. Okay. Yeah. Yeah, so occasionally when I pause, I will have a look here. So this is basically the feather of an ostrich, and when we switch back over again to dark field, and then we're able to see that, it doesn't scatter the light quite as well, so it contrast is not quite as high. Generally with dark field, as you can see in the background, a lot of dust and dirt is visible. That's one of the features of dark field, that it's very dust sensitive. Okay, so I'm going to quickly switch over and check briefly how the vertical live is doing. Okay, because every now and then, yeah, the vertical life also seems to be running. We're gonna see, yeah, which one's more going to be more popular, the horizontal or the vertical one. I don't know. I'm experimenting as always. Yeah. So the ostrich feather is a nice one as well. Yeah, also mostly sentimental value for me. And now let's move over to some of the commercially prepared slides, and they essentially are kind of expensive. some of them, and some of them are fairly old, around from the 1980s. Yeah, and they're made from a German. It's a German company that made those for educational purposes, and the company is also producing slides for medical training. Yeah, so that's the thing. Yeah. Yeah, histology slides from human tissue. I've basically that's an interesting one. If you want to have histology slides from human tissue, generally the company that I recommend, and they have a free online catalog. And you know what? I'm going to just type it into the chat. Okay, so if you want to, you can just visit their homepage and download, even in English, the catalog of slides. Leader.de, so that is a German. Everything's also available in English, so I'm just posting it right now into the chat. Okay, lieder.de. It's a German company. You can download the slide, and they even manufacture slides based on your own how do you say, requirements. Yeah, some of the slides can be quite expensive, therefore, and if they don't have it, I don't know who's got them, honestly. Yeah, so let's have a look at those. This is maize, basically corn. Yeah, the cross section, and yeah. Oh, let me go back to the normal one, and yeah, I need to refocus again a little bit here. Yeah, and that's. Yeah, that's a typical structure of a monocot plant. It's, yeah, we'll quickly also explain a little bit of what you're able to see here. Those round things here, these are the so-called vascular bundles. Yeah, these are basically the cells that carry water up the plant, and also nutrients like the glucose that was made down to the roots. Yeah, so that's the cross-section of a stem, and then in a typical monocot plant, as you see over here, you see that those vascular bundles are scattered throughout the stem here. Yeah, yeah. It makes dark field a great way to check cleanliness of a slide before making it permanent. Yes, it does. However, dark field is so sensitive that even the dirt and the dust that's in the optical path, even that basically you can see. Yeah, so yeah, so that's a little bit one of the things. Yeah. Tini Coco is saying for the long content like this, I prefer the horizontal one. Actually, I have to say, I myself as well. And even if you watch it on a mobile phone, why don't you just rotate your mobile phone horizontally? That I'm fully aware of that. But the reason why I'm also trying in parallel the vertical live is because YouTube also pushes it into the shorts. How do you call this place so that more people are able to actually see it. It's a little bit of an experimentation. Yeah. That's horizontalize my phone. Yeah. Honestly, it's the layout is also much. You can fit way more things if you do it horizontally. So this is one of my favorite ones, and I've actually also made one of it must have been over 20 years ago now, 25 years ago. It was my first microscope. I made my own microscope camera using a webcam. So I disassembled a webcam, constructed myself a separate container, and put it on my microscope. And the webcams at that time had 640 by 480, so tiny. The resolution was miserable. However, I got very good results because what I did is I took overlapping pictures, hundreds of them, and I stitched it together. And this is why I got very nice pictures of those slides. Yeah, yeah. The vascular bundles look like a little bit like faces, like almost, I don't know, like skulls. Yeah. So, by the way, I can turn on the arrow, but not. I think it's not visible on the lives on the short. On the vertical, I mean. Yeah, so those things, it is the vascular, and those large cells in the middle, it's the xylem again, the tissue that transports water up the plant. Yeah. So there is, you see, it's like this. Yeah, of course, there's always a little bit of biology involved, and sometimes it's always nice to know and understand a little bit of what you see. But sometimes it's simply just cool to look at. Huh? I'll give you look. I'm a teacher, and students like to ask questions. And very often there are questions: why are they arranged like that? What's the purpose, or what's the meaning? Why does it look like that? And there's got to be a reason behind it. And I got to be honest with you, I don't know. Sometimes there is no reason. It's good enough. It works. Yeah, the plant is able to survive. So if you have viewed the whole thing from this evolutionary perspective, if it works, then it works, right? And there is no higher reason why the patterns are the way that they are. Yeah? So that's simply one of the interesting things here. So this one over here, that's a nice one as well. Okay, so these here are so-called I show you first the how do you say, always the slide label, so that you see a little bit of what we've got here. These are called Radiolarians. Okay, they have a shell made of silica. So these are the shells of certain microorganisms, and they have a really nice pattern. No, that's not the one. Yeah, here we go. That's a little bit some kind of I don't know, dirt or whatever. Here, this is how they look like. And I'm going to go up now. And here we go. In bright field first. Okay, let's go up again. So these are the so-called the Radiolarians. If you kind of wonder why sometimes the brightness jumps so abruptly, it is I try to pre-program the intensity setting of the lamp of my microscope, but sometimes therefore it starts to jump. Yeah. So that's essentially the different shapes that you've got here. Okay, here we go. So look at this here. Yeah, this is also a typical one where you probably want to do a little bit of so-called a slide stacking. You take several pictures, using different depth, and then you stack it together to get it into one completely in focus image. So there are basically you can try even the free version called Helicon Focus. That's the one that I'm using for stacking, and there is also a free trial for that. Yeah. Okay, so I'm just reading. Would you please add more biology relating to the slides? Pretty pictures alone are fine, but little value. Okay, fine. Yeah. Unfortunately, I don't know where they were collected. Those Radiolarians. Yeah. So that is. But these are basically, also single-celled microorganisms, and that's their shell that they have. Yeah. So those are essentially from water, and they can be found in sand. Yeah, and in certain regions of the world, they're quite common in beaches. Yeah. So you can, there are a whole bunch of different shapes, and they might be a little bit small, but they're quite nice to be also seen under over here, right in dark field. Yeah, you can see them quite well. Unfortunately, I only have dark field with my low power. Yeah. Yes, they are amoeboid cells, and apparently those pseudopodia, those extensions, they are able to extend out of these holes that you see there in the shell. Yeah. See, I have to. I lost the focus now again. Yeah. But you see that those tiny little holes here, right? Yeah. In those shells. Yeah. So they're quite nice. So they're basically marine. Yeah. So and let's go up. Yeah, did you just see the jumping of the brightness? That's because the lamp readjusted. Yeah, that's So foraminifera, they have a shell made of calcium carbonate. And yeah, so that's the thing. So, well, apparently, I'm not so much into taxonomy of those protists, but the problem is that the amoeboid motion, so basically moving around like an amoeba, that is what do we say? It's not a monophyletic yeah, group. So this means amoeboid motion can have evolved separately as well. Yeah, so that's a little bit the thing here, that sometimes structures in biology might have a similar function, but might have a different origin. You refer to this as analogy, right? So let's move on. I'm just on slide number 3, but I've got still so many left that I want to show you, and also some of you want to see a couple of maybe yeah, animal tissue. This one here, that's actually okay. That's actually an interesting one. This is where we actually yeah. See, it's a pathological slide, and I do not, unfortunately, I do not know. I suppose it's muscle tissue, but unfortunately, I don't know which muscle it is. I suppose maybe from a pig, because Trichinella spiralis, that's a nematode worm. And these are basically, yeah, that's a problem, right? So eating contaminated pork, especially, is a problem because then you're eating those worms and the worm eggs, and they're quite interesting to see. Okay, yeah. Okay, you can actually see something quite interesting. I'm reading a little bit. If I'm interrupting myself, then I'm reading. You can get those close to dark field with Olympus. I need to adjust, and maybe also the lower one. There are different ways how I can do that. So the scope view, and I need to get the whole thing in focus again. Okay, and that's muscle tissue, and we're now going to look for, yeah, there are several of these nematode worms which are actually encapsulated in there, and here is already one of them here. Okay, and eating this meat is a little bit, I should have turned, I'm going to turn off the automatic light adjustment here. Yeah. So here we go. Okay, so yeah, I can, you see, with the DIC, I'm a little bit limited here. So what you see over here is this round thing here. Yeah, that's actually a worm that's spiraled up, right? Inside the muscle. Yeah. So what happens is that when you eat contaminated meat, then those worms, they will kind of spread over the blood, and then it will go into your muscle, and they make those cysts in there. And if you've got a lot of those, then this can actually yeah, be a problem. Then you can't move properly anymore. Yeah. So this is a little bit the thing here, and there's been other. I've got the upper. When I change, for example, the upper prism, then you see then how it's able to change the colors. But sometimes it will not invert the colors, because it depends a little bit, I think, on the refractive index, and on the bottom polarization. Yeah, when I change, I can also try to change that a little bit, but it will not go. Obviously, this is not a very good specimen for, yeah, for this here. Yeah. So, but we can go down a little bit with the magnification. Yeah, here, for example, is another one. Yeah, so it's another cyst that you have over here. Yeah. So the reason why I like this so much, this slide, is because you can actually see the muscle fibers quite well, and for this reason, I'm really gonna go in. And yeah, it's here. I'm now at 60. Let's open it up a little bit. Okay. Oh, yeah. And what you're able to see then, yeah, are those long cells here. And yeah, and I think those darker dots that you see scattered throughout, these are the nuclei, because muscle cells are actually quite long. It can be up to 30 centimeters long, one single cell, but they contain hundreds of nuclei. And in the muscle, you have protein fibers that are able to contract, just able to slide past each other, and they're able to contract, yeah, the muscle. So, and if you are very careful, and I know that I've been kind of, how do you say, mentioning this already before, also in other live streams and videos, but if you look very careful, then you're able to see that those muscle fibers, they have those stripes, those fine stripes. They're called striations. Yeah, and those striations are actually a little bit like a test on how good your microscope is. Yeah, those striations are called the light and the dark bands. My students have to know this for their biology exams, and yeah, they're basically very typical of so-called striated muscles. And just one of the, because somebody wanted to know a little bit of biology, one of the things that the early microscopists realized is they have seen those striations. Of course, they didn't know what it is. I just can show them to you, those striations. If you look very carefully, I have to now get the arrow here moving, right? You see that there are those little stripes, going across here, and also over here. And the early microscopists, when they first seen those, of course, they didn't know what it is, but they realized one thing, and that's quite interesting. And I found the original publication somewhere. They realized that, isn't this kind of strange? You find those striations in the meat of a whale, the largest animal, and also in tiny insects. Yeah, you find those striations, those stripes. And that's the interesting realization, is it's kind of interesting, they have the same size. Yeah, so people realized already at that time, wow, on a molecular level, on a microscopic level, the structures are quite similar. And they were surprised that a huge whale, that the size of those stripes, the distance of those stripes is the same as one of a tiny insect. I think it's just in biology, it's a remarkable realization a little bit about the so-called the commonality of life. Yeah, the deeper down we go into the cells, the more similar the living things start to become. Okay, yeah. So that's for this reason I like this slide here. Is not only because it's and again that's one of those pathological slides that you can only get from those special companies that are actually making those. This one, for example, here in the center, that's a blood vessel, a cross section of a blood vessel. Yeah, and you see that some of the muscles were cut longitudinally, others vertically. Yeah, not vertically, traversely. That's the correct term. Yeah. So 2 cysts, but I think there were a couple of more even in here somewhere. It also appears to be a blood vessel somewhere. Yeah. Yeah, so in any case, that is a nice sample of muscle tissue with a couple of those trichinella nematodes, worms, which are basically parasites. Yeah. That's why you should always cook pork quite well, but to my knowledge, all of this pork is tested these days, so it shouldn't be a problem. Yeah. So I'm going to do the following. I'm just occasionally. How does the worm penetrate through the gut membrane? So, to my knowledge, is the following: is that the acid in the stomach sometimes they activates the eggs, and then they burrow themselves somehow through some worms burrow themselves through the walls of the intestine somehow. Yeah, then they're kind of scattered throughout the how do you say, the body. It's pretty scary. Yeah. But that's actually one of the reasons why. Yeah. Oh, here are 2 other cysts in the center. Yeah. So that's actually one of the reasons why raw pork is a problem, or can be a problem, if how do you say, not properly monitored. Yeah. So let's move on a little bit. Gee, this takes me longer than I anticipated, but it's fine. Yeah. I do still have a couple of. This is a nice one over here. What is this? Oh, I took out the wrong slide here. That's not one that I actually want. Oh, okay, it's plankton. Daphnia. Oh, Cyclops and Daphnia. No, that's fine. No, they're fine. These are basically stained specimens of water fleas. One of them is red, the other one's green stained. And let's see. Let's see if I'm able to find them. Dust and dirt all over the place. Okay, let's have a look. Where are they? Let's go. You always want to start with the lowest magnification first. So, here they are. Excuse me, I got a cough. Yeah. So, yeah, I guess I just go bright field here. Yeah, these are basically just what appear to be stained specimens. Where's the other one? Yeah, there were other ones also as well here. Yeah, a permanent slide. They have an exoskeleton, of course. This basically means that yeah, they maintain their shape when you make a permanent mount. Normally, water organisms like algae and delicate water organisms, you want to mount in so-called glycerin gelatin in this mounting medium, which does not completely dry out. But this one over here is completely dried out. So where are the others? Let me see, there were others as well. The green ones, I thought. Okay, no, they seem, maybe it was a different slide. That's an interesting one over here. Ah, here's the green one. Okay. Yeah, I just see that this slide over here is a little bit, yeah. I mean, yeah, I've got some live Daphnia. We've watched them already quite often, but they move around quite a bit. So in that sense, yeah, having them preserved and stained like this sometimes makes observation a little bit easier. Yeah. I wonder what you wrote on the right part of the. Ooh, I did not write anything, but I will just basically show you because you wanted to know very briefly. This is a commercial slide. So I want to show you where this the desk view. So you were kind of wondering about this here, A184. Okay, what does this mean? I just want to show you the original box. Okay, so the original box it looks like this. That's Series A. Okay, yeah, and it's basically it's in German. It says here for schools, 25 specimens. Yeah, so that's a box Series A, and in here, yeah, you've got all. It's all. You see, this is all typed out with a traditional typewriter. And then here the numbers. And because we've got several of these boxes, yeah, they are all numbered. 1, 2, 3, 4, 5, 18, 19, 20, whatever, right? And these are basically the things over here. Yeah. So it says here, yeah, Series A, box A, yeah, Series A, box 18, the first slide. So that's just some kind of an indexing system that we've been using. Yeah. So just put them away here. I do have a couple of antique slides as well that I want to show you. So let's go on. Yeah, these are diatoms. Let me quickly have a look. This one is another one. This one here I've also made yeah. Look, the cross section of the stem. It's huge, and this one is actually a nice one because it shows how the structure of the cells is different to a so-called a dicot plant. Here we go. I mean, this is where it really starts to become artistic. Yeah. So that's a sunflower. Yeah. Yeah, that is really nice. Yeah, and. Yeah, looks quite beautiful. And what we have over here is in a sunflower, a so-called a dicot plant. You have those vascular tissues, the cells that carry the water up and down. You have it on the side, on the outside, right? So, by the way, this is also one of the reasons why you got to be careful if you do not want to damage the bark of a tree, because the bark of a tree, this is where the living material is. This is where all of the vascular tissue is for transporting the substances up and down. And for this reason, yeah, you've got to protect the outside. And in a sunflower, being a dicot plant as well, basically they also have those vascular tissues on towards the outside. So the outside, if it becomes damaged, plant cannot transport the substances easily anymore. Yeah. So this is also one of those nice ones. Let me try how this looks like here. Yeah, and you see, yeah, this is again in dark field. Look, a little bit of dust and dirt here. Yeah. Okay. If you have any green, yellow, or red paper wrapped one, please quarantine them and handle with gloves. The pigments may contain arsenic. I would like to have a little more info on that. That means Does this mean that they have actually used arsenic to make the red color? Is this what you're? Yeah, or did they use arsenic to kind of preserve the paper? Or yeah, I would like to know a little bit more about this, because indeed, I do have slides that are red. So yeah, the arsenic for green. Yeah. Because you're talking about some old Victorian slides, you know what? I will show you those that I have. So that's the sunflower, and yeah, this one evidently is also stained. That's why I've got the blue color here as well. Yeah, yeah, mixed with some of the bright pigments, the green ones. That's interesting. Let me, yeah. So let's do the following. I want to show you now. Let's put this here back. Let's show. I want to show you some of the slides that I bought. I mean, I've already made videos about some of those before, but they are really nice. But again, let me put all of the other slides away. Look at this, what I got here. Okay, so these are some antique slides that I bought, and let me see where the other one is. Look, just put this one here carefully over it. Then I'll quickly, and look, there are some red paper covered ones here as well. Yeah. Yeah, so of course I don't know. So generally I don't like to touch them anyway. I only touch them on the side, also because of the fingerprints and so on. But of course, yeah, you never know what they've included in those. Yeah, simply to also maybe make it longer lasting. Yeah. But these are really not, honestly, these are those antique slides here. Honestly, I think the top quality, really, yeah, and some of them are really 100 years old. So, and we can also have a look at those. So you might wonder a little bit why I put them in a folder like this. Okay, the reason is because ideally slides should be, especially old ones, should ideally be stored horizontally. Okay, because otherwise there might be too much stress on the mounting medium. Okay. One of the other things that I kind of like about those slides is over here, for example, yeah, look at the ring. Yeah, they actually use the color patterns. They made it quite nice. Yeah, yeah. So yeah, so I got this one. Okay, I'm gonna show you now. Yeah, a couple of interesting ones. I should have probably indexed them probably better, but this one over here is a. I just want to show you some slightly controversial ones. I like this one so much, and when I made a video of this, people were not so interested. But this one is has a very high educational value. It's the tail of a monkey. Unfortunately, I don't know what the numbering down here means. I just want to show you that here. And this one is definitely, it's not quite as nice to look at, but it's the most unique one that I have. Yeah, it's a finger of a fetus, of a finger of a baby, of an unborn baby. I mean, yeah, look at this here. The date is not complete, but it says 1800. Yeah. So that is So these are from all of those here. Yeah, one of the more unique ones. However, I've got several others that look extremely pretty. Yeah. So if you want, you can quickly have a look at those two, as well. Let's have a look at the tail of the monkey first. The interesting thing is just if you just hold the slide, you already feel it's quite heavy. Also, the edges are polished. Yeah, so also the glass is of a surprisingly high quality, I think. Yeah. Yeah. Unfortunately, I do not know the year when it was made. Yeah. But the ring pattern here is also similar to the one of, maybe it's from them. Yeah, Arthur J. Doherty, right? He's always making those ringed patterns in here. So maybe it's from the same one. I don't know. Yeah. So the monkey tail. That's that's an interesting one here. Okay, so let's have a look at this scope. And yeah, it's always a little bit out of focus. Here we go. And this is anatomically very interesting because you see all sorts of different tissues and so on. This is an upright field. Yeah. Let's go down with the magnification again because this way we're able to see it a little bit better. What we've got here in the center, for example, that's the cross section of the bone. Yeah. You've got here on the top, I always move it down to the center. These are muscles, yeah, and tendons, yeah, for moving the tail. Yeah. So you've got here on top one of them here. Maybe I will show it to you with the arrow over here. Yeah. So that's here. That's one. Yeah. Set of muscles, and these things here, these are tendons. Yeah, that connect the muscle to the bone, cross section. Yeah. You've got over here. Yeah. Muscles. So it's kind of interesting. You've got basically a pair of muscles that are moving it like this, and a pair of muscles that are moving it in XY direction. Yeah. Here in the center, what you've got here. Yeah. That here is bone of the tail. Yeah, that seems to be the marrow. Again, here, muscle down here, up here, left and right as well. Those round things here, these are arteries, blood vessels. I know it's an artery, not a vein, because of the thickness of the wall. Yeah. Those things that you see over here on the side, that's all fur. Yeah, so it's basically the hair follicles for the fur. Those round things here, the white round things, that's adipose tissue. That means it's fat storage. Of course, yeah, it has to be insulated. Lots of fat in here as well because it protects against cold. So I kind of like this a lot. Yeah, as a histological slide, and honestly, yeah, has survived the test of time. It's still of excellent quality. Yeah. So. So that's for that reason. It was actually a fairly expensive slide. I think I paid around 30 euros or so for just for this one slide. But it's from an anatomical and educational perspective, really great. Yeah. So let's put this away. And the other one that I have over here, that is the less interesting, I would say. You don't see quite as much, but slightly more controversial. It's a human tissue, the finger of a baby, of a fetus. This one over here, that's the fingernail. Yeah, and here again, if you look at it, you see that a whole bunch of muscle fibers and tendons and so on. Yeah, the skin on the outside here. Did the maker of the monkey tail slide mention the preservation method? No. But I just from on that at that time, however, people used Canada balsam a lot as a mounting medium. So I suppose that it was also used. Yeah. So it's a simple histological cut. It was dehydrated, I'm quite sure, embedded in paraffin, and then mounted in Canada balsam. It does not say on the slide, but at that time, Canada balsam, to my knowledge, was pretty much extremely popular. Yeah. So that's basically not quite as interesting maybe. However, you are able to see over here as well. These seem to be some kind of either sweat glands or so. Yeah, those lines that you see that with the little glands which are going out to the surface of the skin. Yeah, so that's it's how do you say, an interesting specimen, but not quite as how do you say, revealing to look at. Okay, so let's have a look again over here. So I'm gonna put it back. There are a couple of others that I wanted to show you. Some nice ones. Let me see if they're over there. Let me see, and what do I have over here? Nope, that's empty. So let's have this one over here. Where are they? Where is this? Entomology B. Where did I put them? Maybe this is still in here. Let me see. I'm already a little bit, how do you say, I've got some vision problems already because I need to get my glasses on here. Where is this? Oh, it doesn't matter. Yeah, let's have a look at this one over here. It's a little difficult to read. Check here. Ah, yeah, here. That's one of them. That's actually one of them that I wanted to show you. That is the this here. That is the cross section of the spine of a sea urchin. A spine is one of those spikes. Yeah, that's quite nice looking as well. Yeah. So let's put it like this. Really nice patterns of nature. Yes. Yeah. Again, if you've been, how do you say, watching some of my videos over the last couple of months, yeah, I've already shown them. Yeah. Before. Looks very nice. Yeah? But again, as I mentioned already before, it's a little bit of a, how do you say, a miracle to me how they were able to do that. And I think what they've done is they've actually glued the sea urchin spine on the slide and kind of polished it down, right? Yeah. So and that's polarization. Right now it's not polarized. Okay, because I pull out the polarization filter. Yeah, and also the prism. Yeah. So right now it's just regular bright field. So we go up a little bit here. I mean, I don't want to get too theoretical about this, but if you're looking at those complex patterns of nature, right, and the complexity of life, there's this concept called an emergent property. Where is all of this information stored, really, right? How those sea urchin eggs, not eggs, sea urchin spines, how they actually should look like, and all of the patterns. Then you're gonna say, well, of course, it's got to be in the DNA of the organism, right, in the genetic material. Yeah, but the problem is you're not able to pinpoint any specific location. Sometimes patterns appear during development. Yeah, and that's the interesting. It's called an emergent property. Yeah, so this is something that also fascinates me a little bit. Is that it's not everything, not all of the beauty and the patterns are directly encoded in DNA, but sometimes patterns emerge during the development. Yeah, so I don't know what this is here. Yeah, let's have a look at it. Yeah, some others. Tongue of a bee. That's I've got also some insect parts as well. Yeah, I mean, yeah, I've got bee stingers, and bee parts are quite fairly common, obviously, as specimens. But I think that this one has been prepared specifically and beautifully. Yeah. So that is basically what you see over here. That's the tongue of a bee. Yeah. I'm always kind of surprised, not only at the beauty of nature, but I mean, this has to be preserved somehow, right? And that it was able, with proper preservation, was able to survive, let's say, 100 years or longer, is quite surprising. Yeah. Yeah. So polarized, there was a request of I can do that. But with those, I've seen it with those prepared microscope slides. Sometimes the effect, the DIC effect, is not quite as big. A little bit here. Yeah. Yeah. So I'm now using polarized light, but this is not just regular cross polarized, but this is DIC. Yeah. So it's here. So the exoskeleton also sometimes of some insects, the exoskeleton is very dark, and therefore has to be bleached and brightened up a little bit. Yeah. It's actually quite interesting because we were talking about some of the more toxic chemicals used in biology. There used to be a solution called Hoyer's solution. Hoyer, Hoyer's solution, and this was used as a so-called as a clearing agent. basically, if you had very dark and dense specimens, you added this, you kind of added this solution, and then it kind of cleared the specimen. There's only one problem with that this solution was very toxic and acted like a drug. Yeah. So people, however, back in the 19th century, they used these things. Yeah. Over time, yeah, sometimes the techniques and methods that were used in the past are simply not allowed anymore. It's simply not good to use them anymore because of safety issues. Yeah? At that time, many people simply didn't know certain things, especially also concerning radiation. I mean, not so much of an issue in microscopy, but back in the day, especially with radioactivity and so on, they were just very often according to nowadays standards, quite careless. But people simply didn't know. Yeah, so that's basically a B. Yeah. Mercury was used. I mean, honestly, before antibiotics came around, people used to administer mercury even as medication. And the reason is because mercury indeed was being a metal, was indeed able to in some cases at least make small improvements because it poisoned the bacteria, right? But obviously also your own body cells, right? So so at that time, that's why I understand when it has. Thank you for the information. If you're saying that kind of, it could be that maybe they're laced with some kind of arsenic or whatever. Yeah, good to know. So let's have another look at a few others. Yeah. While listening to the stream, I'm also looking at the edge of a moss leaf. It's only one cell layer thick. It has little teeth like a nettle emerging. Yeah, honestly, yes. That's one of the interesting things generally is that patterns in nature simply seem to appear, right? And then you ask yourself, well, where in the living thing is this actually, how do they actually know how to make certain things? And then you discover, actually, obviously they don't know. It simply appears out of the way that the different cells interact with each other. So let's go on here a little bit. There are a few other. I think that's one of the things that I wanted to show you. Yeah. That's the ovary of a lily, of a plant, and here this is quite cool because let me show you. Where is it? I have to find it again. I always start at the lowest magnification. Here we go. Okay. So again, it's a little bit dirty, but each tiny little dot is a nucleus. So this is a stained specimen. Yeah, if I change the prism around and you see it doesn't make a huge difference here. Okay. Let's go up yet a little bit more with the magnification. Let's use bright field. And let's see, I don't know, I forgot where it is. Each one of those round circles is of course, how do you say, a nucleus. Okay, so it was stained with a substance. It was stained with a substance that stains DNA, and here we already have the thing. I mean, I already know the specimen, of course. So, and maybe you already noticed something that's in the center here. What do you see over here? Right in the center, right in the middle. It's weird long structures. Well, these are, of course, chromosomes. This is a dividing cell right in the middle right now. Those long structures, these are chromosomes. Yeah. So, there are a few of those over here right now in the center as well. If you, you know what, here I'm going to show you again. Yeah, these are also chromosomes over here. Yeah. So I'm going to show you another one. So that's the reason why I like this specimen here, is because you can actually see cells dividing here. Yeah. Here are chromosomes. This one is in so-called in anaphase. Yeah, the chromosomes have already separated. Yeah. Yeah. This one down here seems to be in prophase. Yeah, so essentially, Yeah, so yeah. Fetal development is a long sequence of emerging properties dictated by gravity and chemical concentration gradients. Yes, and it's a complex process. It's a complex process. They've been studying developmental biology also on fruit flies and Drosophila. Yeah. Yeah, here again, yeah. So you see that that's one of the nice, how do you say, specimens. Now, in case you wonder why some of the cells do not have a nucleus, like over here, for example, you're going to say, where? Why do I not see a nucleus over here? Right? Some cells that seem to be empty, and the reason is, well, of course they have a nucleus, but you don't see it because it was when it was cut, it was sectioned, right? So this means the nucleus, which was above or below, was cut away. Yeah, this is also explains why especially the larger cells, yeah, like the ones that you all have over here, seem to be missing a nucleus more because they're simply larger. The chance of the nucleus being cut away is much higher. And the smaller cells over here, right, they basically are so small that the nucleus is still very much in the center, right? And therefore, when you cut it, you don't cut it away so easily. Yeah, yeah. So that's a little bit. Um, a history of micro technique. Ah, interesting. Okay. Occasionally, what you can do, and I've downloaded as well. There are plenty of all the traditional microscopy books and so on that can be downloaded, and it's interesting in the sense because it kind of shows a little bit how the people at that time, the first observations. And one of the interesting things is especially many of the older books, from the 19th century and so on, when you read it, they're surprisingly, how shall I say that? They're good reading because the people just kind of explained what they've seen. It's not so, it's not quite, often not quite as technical as one might expect. So it was very often those accounts are first-person accounts, what they've done and what they've observed. So it's more like a narrative, so to say, in many cases, which kind of positively surprised me a little bit because I kind of expected the books of the 19th century to be very technical and dry and so on, but quite the contrary. Yeah. So I still remember this one. It was actually written in German about the guy who discovered the first time tardigrades. Yeah, how you observed them, right? And then he basically came to this interesting realization that maybe those tiny animals, they are also able. He was surprised because this one tardigrade was injured, right, and had a little wound, and then it died under his microscope. And then he described that, and he was kind of a little bit surprised that wow, even those tiny animals are able to experience suffering, and he wrote that down in his book. Really interesting how people started to realize, now I'm getting a little philosophical, their own connection to their environment and to the microscopic environment. Yeah, so they saw similarities. And so I think it's quite nice. Yeah. Yeah. So yeah, exactly. There's always something interesting in those. But when you do a little bit of online research, wow, there's lots of stuff. Oh, okay. Here again in the center, right? Some chromosomes. Need to turn up. Yeah. Yeah. Here again. Yeah. So that's also one of the slides that I like, and where I've made some how do you say some pictures, and then I frame them as well and hang them up in my room. This takes me longer than I actually anticipated. I'm not gonna, Okay, that's a more histological one that I show my students, artery and vein. So I'm gonna jump ahead now. Where is this? Here, this is Aristolochia. This is it, that's it. This slide here has a little history. Okay, this is basically the cross-section of a stem of a plant, Aristolochia. It's called just some plant. And it's a fairly large one. It's about 5, 6, 7 millimeters across, right? And what I've done, this was actually one of my first slides where I have taken, I kid you not, a few hundred pictures of this, overlapping pictures using my webcam, homemade webcam microscope camera. And then I manually at that time stitched them together into a larger image. Yeah, just want to show it to you because I think it's pretty nice looking. And that's why I've chosen this one to make those. Yeah, but it's a fairly large specimen, and for this reason, yeah, it's a for this reason I'm not able to fit everything on here. So let's go down again with the, yeah, look at this. Yeah, so here you can also see again this ring-shaped arrangement of the vascular tissues, yeah, which is so typical of a dicot plant. The largest cells again, the xylem for transporting the water. Yeah. So I'm occasionally, as we look at this, I'm occasionally reading here a little bit what's going on in the chat. Okay, and allow me also take a quick look at the other live stream, how the other one is going. In the other live, and the vertical live stream, there are also a couple of people. Yeah, right now. Let me quickly check. Yep. No, at least there are 11 people in the vertical live stream. It's also quite nice. We're gonna see how this actually goes on in the future if people start more people start to watch it then. Okay, yeah, so that is again that's and one of the interesting things what I have done here is I should have actually showed you the picture here is I've taken pictures and inverted the colors, so everything that's white becomes black and vice versa, so it's a negative, right? And this almost looked a little bit like dark field, looks very nice. Yeah, so that's yeah. Question is, why do you have 2 live streams? Honestly, for experimentation. Okay, I just wanted to see how well this actually works. Yeah. Do you know anything about cleaning microscopes? I have a feeling lower end scopes like Swift, etc. need a good clean inside and out to get the best out of them, even if new. Honestly, I wouldn't clean them unless really necessary. Okay, and dust will always accumulate. My general advice is don't clean unless really necessary. Yeah. Magnify and DIC. Okay, let's try the DIC for this one over here. So, and again, probably not always a very, yeah. So let's go up. So that's now 40 times. Let me check here. It's 40 times. I need the correct prism as well and everything. So let's have a look here. Let's focus. And that would be now DIC. And you see, there's not a huge difference. I can play around a little bit with the top prism, right? Yeah, over here. Ah, look, this looks interesting. It's almost, maybe there are some crystalline structures in there. Yeah. Yeah. So the thing is the following. Yeah, dust buildup is, of course, always a problem with microscopes. However, you will mostly only see the dust if it is close to the plane of focus, right? Or maybe on your eyepieces. Yeah, inside, if there's some dust, you wouldn't see it anyway because it's out of focus and blurry. So, and I would really not, yeah, clean. I tell you. What can happen, and I've mentioned this already several times before, is when people are not careful with their oil immersion procedure. So this can be actually a problem because sometimes it can happen that people rotate their non-oil immersion objective into immersion oil, like the 40 times objective. That you shouldn't do that. Then you have to clean it, right? So that's yeah. Are they really crystals? I don't know. It could be. It does look like it. Let's try it again. You see, usually if something starts to light up like this, okay. But I want to show you something else here. I do have yeah, some other slides that they look very much like crystals, but I don't know which ones they are or what they are. Look. I mean, if something lights up like this, then this is and has so many sharp corners, then this is a strong indicator that this might be some kind of a crystal. Yeah. So I'm watching both streams and my brain hurts because there is also a time difference between. Thank you. The questions are here. There's a time difference between the 2 streams as well. Yeah. Yeah. So what I do want to show you now is the following. Okay. I want to show you some other crystals, and this is a microscopy friend from Germany sent them to me a couple of years ago, and this is something I really like to share with you. Yes, I've also made a video already before that, but I really want to show this to you. He sent me 3 slides. I added the labels. So this was back in 2023. Let me quickly go to the desk view again, because these are some really interesting. So that's a water lily. It was mounted in histokit. I don't know that mounting medium. Okay. But these are some cross sections, and I just want to show them to you because they are so-called idioblasts inside here. Idioblasts are cells that also have crystals, and the crystals that are in here are really interesting because they look very different. So, and just I have to find them. There are not that many of them in here, but they are quite nice looking. Here we go. So he prepared those slides and sent them to me. And let's see if I'm able to find them over here. And this one does not seem to have. Okay. So because there are these huge air spaces inside. Yeah, here is one. Here. Okay. Look at this. Do you see this spike thing here? These seem to be calcium oxalate crystals inside the water lily. Very typical. And look, because I'm using polarized light now, look how nicely they shine. Yeah. Basically, without any polarization, this is how they look like in bright field. Yeah. Yeah, and these are basically these crystals that are forming on the inside. And I did a little research because I wanted to know what are they doing. And there seem to be 2 possibilities. Either it's some kind of a protection against being eaten, and or basically it's calcium storage. So if there's too much calcium, then it will simply store it in those crystals here. Yeah. Yeah. So, and let me have another one. Look at another one here. So, I've seen some really nice samples as well. Okay, it's always a little bit. Let's go down a little bit with the magnification so we get a better overview as always. I think there was, you know what, let's go yet further down. Then you have to scan them a little bit, and then occasionally you are able to find them. Maybe if the cross-section would have been a bit thicker, maybe it would have been a little bit easier to see. Oh, come on, where are they? There was another one. Or maybe it was on the other slide, I don't know. Let me quickly check. This one is cut much thinner. Yeah, he cut them at different how do you say, thicknesses. Maybe they're easy to see. Yeah, so I got 3 slides of the water lily. Oh gee. Here's another one. Right in the middle. Yep. Here we go. Yeah, they really start to shine. Yeah. Yeah. So here we go. Yeah. So yep. Let me see. Can I go up yet further? Yeah. Yeah. This is kind of nice. Nice looking. Yeah, again, these are so-called these are crystals, and the cells that form them inside the water lily are called idioblasts for whatever reason. Yeah. So idio means different. Yeah. So these are cells that are kind of different. Yeah, and that's also something that I just wanted to show you here. Let me quickly check. Again, I've got a couple of questions here. Yeah, I need to check a little bit some of the questions, and maybe I'm going to show you what time is it. A little bit, some more of the things here. What else was there? This one here. Yeah, that's another favorite one of mine. Yeah. This one actually, I kid you not, this one was featured in a BBC documentary a couple of years ago. The BBC contacted me because they found the picture of this flea somewhere on my webpage, and they asked me if they can use it for documentary. And I said, of course you can use it. And then later on, they actually sent me the file, and yeah, this was actually featured in a BBC documentary a couple of years ago. Yeah, so I was quite happy about that. Yeah, and so yeah, so that's the flea of a dog. Okay, yeah. No, I would not have charged them. Being allowed possible to contribute to BBC documentary is an honor. Yeah, it's yeah. Then I got the MP4 file, and it is indeed about microscopy. Yeah, about technology and the microscopic world and so on. It was a whole series. I've forgotten now the name of it. But the thing is it was basically also very suitable to be shown to my students. Yeah, yeah. So if we zoom in here, that's the other thing that I want to do here. Look, this also looks quite nice. And what do we see again here inside the flea? Again, the muscles, right? And again, you can see those stripes that we talked about. Yeah. So you see that. Yeah, there is the common, as I call it, the commonality of life. Again, you have those light and the dark bands inside the muscles here. Yeah, so it's quite a nice one. Again, this one was actually also one of those specimens that are a little bit too large to be, yeah, so I had to also stitch them together to make a nice picture. Yeah, so usually when making those hole-mounted slides, they're not so difficult to make, really. But the problem with them, as always, is that in many cases the specimen is too dark. It won't allow any light to go through, and then you don't see any details. So in many cases, those insects, they have to be bleached a little bit. There are a variety of ways how you can do that. Potassium hydroxide, for example, is quite common. You can use lactic acid apparently also to bleach them. Never tried that though. And then some mounting media, like for example, Uperol is also suitable for brightening them up and clearing them. Okay. Yeah. So I'm going to quickly. You know what? Instead of showing you the how do you say the questions, which you can read anyway in the chat, I'm just going to read them. Yeah. From over here. I would like to get some prepared slides of human histology samples and would like a reliable supplier. If you really want to have a reliable supplier, I really recommend that you contact LIDA. I typed in the link in the chat. It's a German company. You can download their huge slide catalog. But let me tell you, they're not the cheapest ones, okay? But they're high quality. Yeah, so they're making those slides also for medical training and so on. Yeah. Yeah, the comment that basically I should handle some of the slides with gloves because of possible poisons which might be on the paper. Yeah. Yeah, dark field, a great way to check for cleanliness. That's possible. I would say the following. Look, when you're taking a natural sample, a pond water sample or whatever, I mean, there's so much dirt in there, dirt under quotation marks in there in any case, that sometimes it doesn't make a huge difference if the slide is perfectly clean or not. Okay? I'm just saying the biggest contamination under quotation marks actually sometimes comes from the specimen itself. Yeah? So, Where is the link? The link I typed in the link to the company into this chat. If you want me to, you know what? Whoa. Again, I'm gonna write it down. I'm not affiliated with the company. Okay, I'm just saying. Yeah. So I'm not working for them. I'm not affiliated with them or anything. Yeah. But in case you want to, let me see. I need a pen. Where's a pen? Okay, if you let me write this down on here. Yeah. L-I-E. Okay, yeah, lieder.de. Yeah, if you want to have cheap microscope slides, permanently mounted microscope slides, buy them on Amazon. Okay, that's of course the yeah. Buy it on Amazon. However, I do have to say that those cheap Amazon slides, I mean they work, right? But quality-wise, generally there are no comparison. Really, yeah. So it's not a comparison. Yeah? The quality is much lower, much more dirt. The Amazon depends, of course, which brand and so on. Obviously, Amazon is just a retailer, not the company that makes them. But because they're much cheaper and so on, there is quite a lot of dirt sometimes. Yeah. AliExpress has some surprisingly good ones. Okay. Yeah. So again, my suggestion is as always, don't spend too much, and yeah, take it from there. Yeah, let's we said is we want to look at a few things here. Okay. So some questions that I already answered about the nematode worm. Yeah, then the question about the slide labels. Does anyone know of companies that make permanent slides of different fungal tissues? Honestly, not that I know specifically that they make that, but if this company doesn't have that, then then I don't know who's got it. Yeah. Some of my favorites that I managed to get, I have 2 slices of rabbit fetus head. Wow. Okay. The side view, you can see the teeth forming in the gums. Rabbit fetus heads. Wow, that's an interesting one. Okay. The monkey, the maker of the monkey tail slide did not mention how the slide was made, unfortunately. That's always one of those problems. In last week's streams, when you showed the viewer the use of the 2 polarizing plastics, was it still in DIC or doubly polarized? When I polarize the plastics, no, what I've done is briefly, if I remember correctly, let me show you. No, that's the wrong one. Desk one. I've got some polarizing filters here. You can use plastic filters, and basically what I've done is I've simply placed it on how do you say, the one on top of the slide and one over the lamp, and I removed everything else. Right. So it was just basically cross polarizing. So, and that's essentially something that can be done quite cheaply. I would probably not buy those glass ones. I got them from very old camera filters. Yeah, but they are extremely cheap polarizing plastic filters. Yeah, yeah. Okay. A dual comment from Ever Smaller. Dual biology slide stream at some point, but never got around to arranging it. Yeah, that would be actually quite an interesting thing. It would be kind of nice to have some kind of a little bit more interactive live streams where maybe it would be possible for different people to kind of show at the same time what they've got. It would be quite nice. Yeah. Okay. Yeah, I'm just going through the questions here. At least those that I did not mention yet. About the 19th century book. Internet Archive is a great resource for a very old. Yes, that's correct. That's also where I got mine. It's called Internet Archive, and it contains basically scanned books, and you can read them directly on screen, or you can download them. They're all in public domain. yeah, because the copyright expired. Yeah. So that is something that I also recommend. But then again, it it takes a lot of reading to do. Yeah. So essentially, then, but it is possible to search also for terms then, because it's indexed, and this also makes it then easier for you to find the interesting things inside those old books. Yeah. So let's move on a little bit. What time is it? Okay, in 10 minutes or so, I'm just gonna call it quick. A day again, show some 10 minutes. Your DRC has a button to turn your DRC polarizing on and off. Let's put it this way. Well, it's not a button, but it's a. What I can do is I have several possibilities. I have a, and I cannot show this to you. Well, actually, maybe I can, but it's gonna be upside down. So let me see if I can. If I can do that, yeah, you see, it's upside down. You hope you don't mind, yeah. So if I want to switch the DRC off, I just remove the polarizing filter like this. Okay, yeah, that's one thing that I can do. So that's adds the polarizing filter, and then there's another thing that I have to go up here. Up, does this work? Yeah, there is this, and this, which you cannot see because this here is my, by the way, this thing here at the top, this here at the top, yeah, that is I glued the camera on here so that you're able to see the stage here, right? And there is this thing here that contains a prism. Okay, I mean, if you are interested, I can take it out and show it to you. Yeah, it's called the Wollaston prism. Yeah. And by turning this here, yeah, I'm shifting something, and then this is able to change the colors. So how do I get it back? Yeah, in case you just were kind of wondering a little bit of how this actually, yeah, how this actually works. Yeah. Yeah. Okay. They have their own abbreviations. Okay. Yeah, this one is another nice one over here. This one is wood. Yeah, in different sections, and there's also one of those slides where you can make pictures. So actually, honestly. What I recommend is if for those folks who are starting out with microscopy, just get yourself a box of slides because it's almost, as I say, almost nothing can go wrong, because especially if the slides are nice looking, they look extremely artistic. Here, for example, this one is also one of the things that where I've taken the picture and where you can hang it up on your living room wall. So again, the individual cells here, and regular bright field. You see how much light it takes away. Yeah, it looks like this. Yeah. Wow, you've got 1,000 slides, really? Honestly, that's way more than what I have. Seriously, that's way more than I have. Usually, what I usually do is I make, of course. A lot of slides, and then usually I film them or I take pictures, and then these are just temporary slides, and then I simply wash them. And what I usually do is the following. Just a second. I always dump the slides into a plastic box like this, right? And then I wash them. I pour hot water over it, and then I wash each one individually, right? So I'm reusing those. Yeah. So just that's basically how I do most of my microscopy work. Put this away. Okay. Yeah. So, and maybe one of the last slides I want to show you. This is a huge one, and honestly, man, making this. Look at this here. Yeah, it's That's the flower, female flower of a pine. Yeah, and looks also very beautiful. And yeah, the complexity of nature really can be seen here. And this is actually also where I've taken a picture and made a picture and put it up on my living room wall. So where is the scope view here again? Need to refocus it a little bit. Yeah. Also properly stained, properly prepared. And those round things here, I suppose, must be somewhere. There must be also the ovules, which will then develop. Yeah, I think this one over here, which will then develop into a seed. Yeah, so occasionally, if I'm quiet, then you know that I'm reading the chat. Yeah. So one of the nice things, because you're talking about slide collecting right now in the chat, it's one of the nice things also about microscopy. You can essentially do it in different directions. I mean, you can go into collecting slides, historical slides or modern slides, doesn't matter, or people like to sometimes do microscope maintenance and repair them. Others like to use it more for casual nature observation, collecting samples in nature and the environment, and then just seeing what's there. Yeah. So it's a little bit of how do you say? If you allow me to get again a little bit philosophical, it's. How do you call this? I'm missing now a little bit the English term here. It's like a counterpoint to the fast-paced online world that we have these days. I mean, I know I'm making videos right now, so it is online. But as you've said, it's to a certain extent a slightly meditative hobby as well. Yeah. So yeah, in that sense, it's quite I would say grounding under quotation marks, if this is the correct term to use. Yeah. Yeah, of course, here we again, every week, I know you're already starting to feel tired of that. What is this? Those those vessels that have these ring-shaped structures in the cell wall. Again, the xylem. Yeah. So there's some comments here. What I use for ringing slides is I found indeed nail polish to be quite effective here, but you have to thin it down a little bit using acetone. I think very often the fresh nail polish is still very thick, so acetone. Or nail polish remover, a couple of drops to the bottle will actually make it flow better. And then sometimes when I try to make my slide rings, I don't use a brush, but I actually use a pipette. Yeah. Yeah. So this is basically again also one of my favorite slides here. Okay. Yeah. Okay, what I'm going to slowly do, it's been now again approximately 1.5 hours. I too start to feel a little bit tired right now, talking, and yeah, and anything microscopy is interesting. Yeah. I have to say the following. These days, luckily, it's easier than ever before because to do microscopy as a hobby because the devices, the microscopes are easily accessible and also affordable. I mean, this used not to be the case. I remember when I was still, I don't know, 30 years ago, just finished university. I wanted to get myself a microscope, but they were expensive. I mean, and they were not quite as readily available. Yeah. So for this reason, these days it's significantly easier to simply start it. Yeah. What would be the best mounting medium to secure 5 specks of shiny metal in deep slide? Oh my gosh, a shiny metal in deep slide. I mean, generally, I don't know what the best is, but what I usually experiment around with is before you get any special mounting medium, to use PVA glue. It's water-based. Yeah? This seems to work quite well in most cases. Yeah? Just want to show you. Yeah, you can try to use glues. I'm just going to hold it up like this here. It's clear glue. It's Elmer's PVA glue. It's clear water-based glue. And maybe some of you also know this one over here. Yeah, it's not the acetone one, but the water-based one. Yeah. So that is... No, that's the wrong one. That's the regular one. There is a one with a green label called free of solvent, solvent free. So yeah, that's basically water based, and you can try. Yeah, yeah. Yeah, refractive index won't be too important. Correct. Yeah. Experiment, I would say. Yeah. So what I'm going to do now, folks, is I'm just going to call it again a day. I want to thank you that you joined in again. Hope that you liked it today. I just wanted to show you a couple of permanent slides that I collected over the years, some of which you've already seen, some of which maybe not yet. I'm going to call it a day today, and hope to see you again next week. And what I'm going to do then is I'm going to see how many people actually watched the vertical live stream, and also to what extent that actually worked out or not. We're going to see. In any case, from my side, I just want to wish you all the best. As always, happy microbe hunting, and hope to see you again. And bye-bye. Have a nice weekend.